more DNA. We're going to talk a little bit about organelles. And then next week, depending on how far we get today, we're going to start on mitosis. All right, so yesterday, Wednesday in class, we began to put our nucleic acids together. We looked at nucleotides. We looked at what, if I say draw a base, right, what that means versus draw a nucleoside versus draw a nucleotide with some number of phosphates on it. All right, you should be comfortable with any of those terms. I also want to clarify one more time as far as drawing these structures, what I expect. I drew nucleic acid one more time. A little more caffeine in my system, so hopefully fewer mistakes. All right, so there is nucleic acid, a dinucleotide. Notice this time, all right, for ease of drawing, you can omit the carbons. I'm going to assume anytime there's a node between two lines that you mean there's a carbon there. Right? We talked about how for the carbons you don't have to draw all of the hydrogens, but you do for any functional group. Right? I got the hydrogen on my guanosine this time. To get all the bonds on there, pay attention to the hydroxyls. For the phosphates, because these are in aqueous solutions, we're drawing them charged for this class. And I believe that in recitation, at least a few recitation sections yesterday, there was some question about that triphosphate, whether you had to draw it vertical or horizontal. I'll accept either. You'll see it more commonly drawn horizontally. I find that easier for me to get drawn out properly, but either is fine. And you have to draw out all three phosphates. I have a question about that in lecture on Wednesday. You can't draw one and then say times 3. All right? You have to draw them all out. Okay, is that clear? Are there any questions on what I expect as far as drawing the nucleotides and nucleic acids? Is that DNA or RNA? Why? Got the 2' hydroxyl. All right, good. All right, so hopefully we're all on the same page there. All right, so this is where we ended on Wednesday. We put together a single strand of nucleic acid. For DNA, we started to look at putting together double strands. That's held by what kind? Held together by what kind of bonds? Hydrogen. Hydrogen bonds. All right. So this has some important implications for the way that base pairs pair up. For homework, you had to think about Chargaff's rules, which are based on this hydrogen bonding between base pairs. So what are Chargaff's rules?
Adenine goes with thymine, guanine goes with cytosine.
And they come in equal amounts.
Adenine goes, okay. Which equal amounts of what?
ANT at equal amounts, GNT at equal amounts.
Slower. That came out very quickly.
ANT at equal amounts of each, and GNC at equal amounts of each.
Equal amounts of each. Why equal amounts?
They have to be paired with each other.
Because they have to be paired with each other based on our hydrogen bonding. Right, so there's one higher order rule that comes out of this. Adenine and guanine, what are they? Purines equal pyrimidine, right? Pretty straightforward. So if you understand that, you shouldn't have any problem. Right? If I give you the percentage of any base See? Sorry about that. Not sure that I can move. What's going on here? There we go. All right, so if I give you the percentage of any base, you should be able to tell me what the percentage of any other base is in that genome. All right, simple math based on Chargaff's rules. Right? And these are the rules. You can use those to calculate the percentage of any base if I give you one of them. All right, everyone have a chance to respond? Take a look and see what we think. All right. Overall, good job. That is the correct answer. I think you've got Chargaff's rules figured out. All right, pretty straightforward. Hopefully, if you have questions, ask practice and recitation this week. All right, so base pairing and those hydrogen bonds have implications for the content of each base in the genome. It also has implications for the structure of the double-stranded DNA. So if we look now a little more detail, rather than the cartoon I just showed you, at the structure of DNA, what do you see? You see grooves. Well, we don't see grooves yet. We will when we twist it up. We're getting there. Difference in the number of hydrogen bonds. What are the number of hydrogen bonds between A and T? 2. G and C?
3.
What else do you see? The backbone? Right, so that if you look at the backbone of each single strand, that sugar, phosphate, sugar, phosphate, sugar, phosphate, that we said had a 5' to 3' orientation, they are opposite each other in the two strands. We call that anti-parallel. They go the opposite way. Right, so the 5' end of one strand is on the opposite side of the 5' end of the other strand. Anything else worth mentioning on this one? All right, so this is what it looks like stretched out. That's not the form, native form that DNA takes. DNA wants to twist up into a double helix structure. All right, now what do we see in this one? Grooves. Yes, we're getting the grooves. All right, getting in the groove. What do we notice about the grooves? There are two types. What do we call them? Major and minor. Why are there two types? Bond angles. Very good. All right, so we're going to take a little bit closer look at that. Before we do, remember we're concerned with dimensions in this class, how big things are relative to each other. For homework, you had to figure out the diameter of a double-stranded piece of DNA, 2 nanometers. Other important dimensions that I'm showing on this figure, 1 complete turn of a double helix, 3.4 nanometers. It takes about 10 base pairs, or there are 10 to 11 base pairs in one complete turn, which means each base pair has a height of about 0.3 nanometers. Some other things to notice about the DNA structure. How many ways are there to twist things up?
4.
Oh, really? Well, if you take two things and twist them right or left, right? The native DNA in our cells has a handedness. And it can oftentimes be hard to tell, but it's fun if you go out and look at the way DNA is drawn in popular science places. They often get it drawn the wrong way. So, all of you guys, however, shouldn't do that, right? So DNA is a right-handed helix. You can tell which way a helix is turning by looking at the strand of DNA as it comes over the top. Which way is it going up? So this is going up and to the right. Up and to the right. So that's a right-handed helix. Left-handed helix, up and to the left. Surprising how many ways you see left-handed DNA helices drawn. All right? The native conformation of DNA is right-handed. There are some forms, rare forms, that it flips the other way. That's pretty rare. Right? Generally, it's in that right-handed conformation. Why does DNA want to form this double helix twist? Forming those grooves. Anybody know? Bonding glues is what gives us the grooves. What gives us the twist? Base stacking. Interactions between those sets of bases. All right, bases are happiest when they're stacked right on top of each other. That has to do in part with the different components of our nucleic acid strands. So the bases, hydrophobic or hydrophilic? They're hydrophobic. So they want to be on the inside of the structure. The sugar phosphates, hydrophobic or hydrophilic? Hydrophilic because of the negative charges. Right? So they're all right on the outside. All right, so that's one thing driving this twist. What about, so we have some more dimensions, 6 angstroms between our sugars, right? If we look at how much of that is base, 3.3 angstroms versus the phosphate hole between bases, that hole is just big enough for water to fit through. But our bases are hydrophobic and don't like water. So by stacking and twisting, we essentially get rid of that hole. All right, so it's the interactions between the different components of the double strand, the bases versus the backbone, and trying to get rid of that hole that water can fit through, that drives that twist. Those bases want to be stacked right on top of each other to get rid of that hole. All right, so we're driving the twist because of the stacking interactions between the bases. Now we get to the grooves. So if you look at that double-stranded helix, there are 2 types of grooves, major and minor, little and big. Why do we care? Where do proteins bind? Generally in the major groove, right? So we're going to start talking about proteins and interactions with DNA. There's more space in the major group to bind, number one. But there's also a lot more information content. So let's take a look at that. What do I mean by information content? So if you're a protein that wanted to bind a specific sequence of DNA, some recognition sequence, what do you need to be able to do? Recognize that sequence, right? How is the protein going to tell what bases it's dealing with in what order? All right, so if we look at our major and minor grooves, so first of all, the bond angle, right, that glycosidic bond, is what's driving the size of those grooves. Major groove, minor groove. What do you see in each of those grooves as far as the bases are concerned? What's sticking out? Some of our functional groups. All right? So a protein is going to bind based on interactions of hydrogens. So we can code these functional groups based on whether they are just a hydrogen, a methyl group, or hydrogen acceptor, or hydrogen donor. All right, so if we start here, what about this nitrogen? Is it an acceptor or a donor? Or it's hydrogen? Okay, heard it over here. Does it have any hydrogens? Therefore, it has to be. What about our oxygen? Does it have any hydrogen? Nitrogen. There we go. And then we have a hydrogen, right? So we have an acceptor, acceptor, donor, hydrogen. So there's a pattern based on the functional groups that are exposed in those grooves. What about in the minor groove, nitrogen? Amine? Oxygen? So you can go through for each base pair combination in each orientation and figure out this pattern. All right? I should put 240, 120, just to give you an idea of the difference in those angles. All right, so if you do this for each combination, laptop. What you get is a code. So on the left, adenine, thymine bonds, hydrogen bonds in both orientations. On the right, cytosine, guanine in both orientations. And then the code for what's exposed in the major versus the minor groove for each of those orientations. Tell me what you see. So for the minor groove, what do you see between A and T versus G and C? Repetition. Donors versus regular hydrogens. What about for the major groove? There's no what?
There's no other hydrogen in the major.
They're reversed.
There's no hydrogen, they're reversed. So the fact that it's reversed, what does that mean? Right? In the minor groove, it's the same in both directions. In the major groove, it's different.
You can't tell the minor group.
In the minor groove, you can't tell what?
The difference between the exposed or TA or AT.
Right. So in the minor groove, there's no way to tell orientation. You know that it's an AT base pair, but you don't know whether it's TA or AT. The major group has more information content based on those functional groups that are sticking out, or hydrogen. Right? It's different in the different orientation. Means a protein looking for a specific sequence can identify where to bind and on what strand. Right? We need to pull it apart. Major and minor grooves. Twist is driven by base stacking interactions that give us those grooves, and there are important differences between them as far as interactions between DNA and proteins. Yeah, question?
Your mic died.
Can you guys hear me in the back? Can I shout loud? All right, I can turn the mic on down here. Thank you. I'll stop wandering. All right, so we're getting into the nitty-gritty of DNA and structure. What are some of the implications that this has for the way DNA behaves in solution? All right, so thinking about what you're doing in lab, I think you have PCR week coming up soon. Okay? If you're not familiar with PCR, well, you're going to have to think about it for your homework, first of all, so that'll help get you prepped. But these hydrogen bonds and the structure have implications for things like DNA denaturing. So melting apart those two strands from a double strand into single strands. All right, so we can profile how double-stranded DNA denatures by making a melting curve. So temperature along the x-axis, relative absorbance, which tells us whether or not those strands are in a double helix structure or a single helix structure. And we define the melting temperature as the point at which half of our DNA is melted apart. So half has become single-stranded. If we think about our hydrogen bonds, what does the content, nucleotide content, have to do with our melting temperature? Yeah, more specifically, what does the GC content have to do with melting temperature?
GC has one more hydrogen bond on AC.
All right, so what does that have to do as far as the stability of a double strand?
GC is more stable than
More stable. Takes more energy to melt that structure apart. So if you have a genome that is high in GC content, it's going to have a higher melting temperature. What sorts of organisms would you expect to have high GC content genomes? Thermophiles, things that live in really hot environments, tend to have genomes with higher GC content. That's basically what we just said. More GC, more hydrogen bonds, higher melting temperature. What about reannealing? So if we melt apart a whole bunch of double-stranded DNA, can we get it all melted apart into single strands? And then we cool it back down to reform those double helices. So we know that there's variations in melting temperature. So the GC content is controlling the way that double strands melt apart. What do you see about the curve? So here's our melting curve. What about our reannealing curve? Takes a lot longer to happen. What about just the shapes of the curves?
More gradual.
It's more gradual. What does that tell us about the process?
it's easy to break it down, but it takes more time to put it back together.
Easy to break it down, more time to put it together. Any other ideas? It tells us that something different is controlling the than just 2 versus 3 hydrogen bonds. All right, so if you think about a bunch of single strands floating around in solution, what has to happen for them to reanneal? They have to find the right match. Right? So DNA concentration, more than GC content, controls reannealing kinetics. Because it's about finding the proper match to get reannealed. All right, so we can think about all of these things relative to the structure of DNA and begin to think about them more comparatively. We can look at them, for example, we can look at reannealing, concentration of DNA over time in different genomes. E. coli bacteria versus cow. So you see 2 different curves there, meaning that they're reannealing at different times. So there's some other factor controlling reannealing kinetics as well. So take a minute, discuss with your neighbors. What else? Why do we see this difference? What are some ideas about what's going on in the genome that's controlling our reannealing kinetics between these 2 species? I see a lot of people looking at each other. I don't know. Let's see what you guys came up with. So what did you come up with? What else is driving reannealing kinetics if we're looking at different genomes? Concentration of. So is there another way to say that? Think about E. coli versus cow. The length of strands. Take it one step further. What does that equal? number of base pairs, genome size, right? I think, which is what you guys are all saying in different ways. So genome size is going to control this, right? How complex it is. What other things might drive some of the differences here? DNA structure, plasmid versus, well, they're both double helices, but plasmid versus chromosome, sets of chromosomes. That also plays a role. What else? So there's differences in the sort of information content versus length of the piece of DNA and how they might be able to match up quickly or slowly. That plays a role. All right, so far, most answers are centered around the size and shape and amount of DNA. What about content of the genome? What sorts of things do we find in genomes? Repetition. So lots of repeated structures. What else do we find in genomes that make us work? That are expressed and make proteins and genes. Thank you. There are genes in genomes. All right, so there's lots of different types of material of DNA in genomes. And different genomes have different proportions of these various categories. All right, so the genome structure, the content, plays a role. So if you're trying to reanneal things that are highly repeated versus non-repeated, right, that controls differences in kinetics. So looking at these reannealing curves can give us a sense of what's going on in genomes. In general, a very small percentage of our genome consists of pieces of DNA that code for products, whether that's a protein or a piece of RNA, ribosomal RNA. Right? 2% are coding. The rest of our genome is either regulatory or non-coding stuff we don't understand or repeated structures. And the various amounts you have of these different categories are going to control some of those reannealing kinetics. All right, so we've thought about DNA. We've thought about how it goes together and the structure, how that structure affects things like denaturation and reannealing, how that relates to genome structure. The next thing we're going to start moving into is thinking about organelles. All right, we want to think about, we're at DNA now. For humans, we took a human chromosome, stretched it out end to end, it'd be about 2 inches long. All right, so maybe not that long, that long. But that's a pretty huge amount of something to fit in a cell. All right, so the first thing we want to talk about is how we package our DNA so that it fits in the nucleus. All right, so we'll end today with just a few quicker questions to get us prepped for talking about organelles first thing on Monday. All right, so this should be from your homework. So it should be completely out of the blue. We're packaging up our DNA. It's wrapped around some stuff in a structure.
But it never asked for the name of the filter. It just was like, what is it? I know, but that's the thing. Yeah. Only the single and the
Looking for the specific name. It's one of those. Right? Not asking for what the proteins are called, and asking what the entire structure is called. Let's take a look and see what you think. All right, so People tend to lean a little more towards the octahedral complex. The answer is actually nucleosome. Right? Nucleosome is that set of proteins that the DNA is wrapped around. Alright, that's bead structure. Alright, how many bases of DNA does it take to wrap around those proteins? Remember, we're concerned about sizes and dimensions of things in this class. And get your answers in. Two base pairs. Let's take a look. Alright. So a bit of confusion, not sure. The answer is 150 base pairs. Alright, it takes 150 base pairs to wrap around that set of 8 proteins. Alright, here you go, whoever was shouting out for the first question. Within the nucleosome, what is it composed of? What are the individual units? Alright, everyone should have this one. Your answer's in. Take a look. Alright, excellent. Good job. Histone is correct. One more, I think. What kind of macromolecule are they? Everyone better get this one.
Okay.
Get your answers in.
Maybe? Yeah.
Take a look. All right.
That sounds right.
Good. They're proteins. All right. So all of these questions are prepping us for looking at how DNA is passed into a cell, and passed into a nucleus to be more exact. All right. So we're going to pick up on Monday, looking at DNA passing. Move into organelles. Everybody have a good weekend.